Thank you, Matt, for nice and detailed introduction, and thank you all of you for being here today. So as Matt said, today I will present an overview of the research I've been conducting at Eugène's lab during the past 7 years. And this is how I structure my presentation for today. I will start with a brief introduction, then I will move to the two main components of my research. Then I will spend a couple slides talking about education component that result from this international research collaboration I have been involved, and I will close with a brief summary. So as many of you already know, wheat stem rust is caused by the fungus Puccinia gramineae speciale triticide, and from now it's going to be PGT. It's a pathogen not only on bread and durum wheat, but also cause disease in barley and triticale. And it's one of the most devastating diseases of wheat, which can cause completely crop failure in highly infected fields. Wheat stem rust can be controlled by the use of genetic resistance, fungicide application, which is a common practice in Europe and also in the United States when the prices of wheat are high, and also by the eradication of the alternate host. And this has two main impacts. The first one by reducing the initial inoculum from the barberry bushes into the wheat crop, and second and most important, by eliminating the sexual stage of the pathogen, so drastically reducing the number of races that can occur through sexual recombination. So wheat stem rust has been effectively controlled by the use of genetic resistance, in particular during the second half of last century. So some affecting genes, most notably SR31, play a critical role in controlling stem rust all around the world. But this story changed since the occurrence of UG99. UG99 was first identified in Uganda in 1998 and was technically designated as race TTKSK following the North American letter code nomenclature system. So why this race was so relevant? First, UG99 was virulent on over 80% of the world's deployed wheat varieties. It also has been rapidly migrating and evolving into new variants. And some of these variants also gain virulence to other resistant genes that are widely used in agriculture, particularly SR24, 36, and TMP. So this is the original UG99, found in 1998, and you can see its relevant feature is this virulence on SR31 and also in SR38 that is not present in this table. As you can see here, in a matter of 17 years, 11 additional variants in the UG99 has been identified. And what's more relevant, some of these variants, like for example TTKST, has combined virulence to highly important genes, in this case 31 and 24, TTTSK with virulence combination on 31 and 36, and lately the most virulent of all, the TTKTT, that combined virulence on 31, 24, and SRTMP. And also the variant of UG99 has been rapidly migrating. You can see from this map, in 2005, only 2 variants in the UG99 group were described. TTKSK, at that time present in Uganda, Kenya, and Ethiopia, and race TTKSF, present in South Africa. But if we go all the way to 2014 and 15, we can see that all the countries in East Africa has at least one variant in the UG99 group. Also, UG99 has been identified in Yemen and Iran in the Middle East, and has the potential to migrate and reach the high production wheat areas of the Punjab in India and Pakistan. So in response to the threat posed by UG99, first the Burlock Global Rust Initiative, or BGRI, in 2005, and then the Durable Rust Resistant in Wheat Project, or DRW, in 2008 were established. And the DRW, it's a comprehensive global program that congregates 21 research institutions around the world with the main objective of reduce wheat vulnerability, not only to stem rust, but also to the other wheat rust. So they were coordinating activities aiming to replace the rust-susceptible varieties with durable resistant ones. And 2 of the relevant objectives under the DRW umbrella were, first, increase the stem rust surveillance to detect and monitor not only UG99, but also other significant races. And second, persistent discovery and breeding to develop stem rust resistant varieties. And in these two activities is where I have been working in the past years at the Cereal Disease Lab, and result from this research is what I will be presenting today. So let's start with stem RAS race typing. So one of the main responsibilities we have at the Cereal Disease Lab is to conduct the National Stem RAS Survey. So every year between 100 and 150 stem RAS samples coming from all the wheat growing regions in the United States are received and race typed at the Cereal Disease Lab. But in addition to this national effort, we also have an extensive collaboration with international research centers, focuses on race typing of international samples with the objective of first, monitoring the origin and spread of new virulence in the pathogen, and second, to determine the degree of pathogen variation in particular countries or regions. Just for you to have an idea, these are the countries from where we have been receiving and processing samples in the past 5 years. And even though our focus is been mostly East Africa and some areas in the Middle East, you can see that we have been also processing samples from Asia, Europe, and also South America. So every year we receive between 250 and 300 samples from abroad. And these samples, when they are shipped between November and February, they are processed here at the Cereal Disease Lab. And we have a time restriction to process these samples. We can only work in the winter months, starting in December 1st, going all the way until March 31st. So what happened with the samples that are collected and shipped outside of the winter season? So we do have a support laboratory, the Foreign Disease Weed and Science Research Laboratory in Fort Detrick, where the samples that are shipped between March and October are received, and those samples are increased. So when we reach in the winter months here in St. Paul in December 1st, this laboratory shipped the sample to the Cereal Disease Lab when we can process and race type them. And this chart shows the methodology that we follow in order to analyze these samples. And due to this time restriction, I would like to say that in order to accomplish all these steps, this is only possible because the really good interaction and the 3 stem RAS laboratories at the Cereal Disease Lab we have. So Eugene's lab, Les Sabo lab, and Matthew Rau's lab all working together, aiming to accomplish all these steps. So the first thing we do when we receive the samples is race type them based on the North American differential set. And these are 20 resistant genes, and the result from them is this 5-letter that we race type each of the race. In addition, we characterize these races on a set of 26 wheat and barley resistant genes. And this is really important because it provides us a broader picture of the virulence spectrum of these races, in particular to critical genes that are not present in the differential set. Once we characterize the races, we handle them to Les Sabo. When they come at their lab, they perform different genotyping assays. And finally, whenever significant new virulence combinations or races are identified, we incorporate them into our routine SharePlus screening. And this SharePlus screening does not only include the North American winter and spring wheat nurseries, it also includes the international nurseries mostly coming from CIMMYT, and also nurseries that are coming from the countries where the races we are evaluating are relevant. So in the next few slides, I will show a couple of examples of international surveillance efforts where the CDL has been heavily involved, and I will share with you the type of result that we are able to generate here in St. Paul. So let's start in Ethiopia. So in addition to the threat posed by UG99, since it was discovered in 2005 in Ethiopia, in the country there was a severe stripe rust epidemic in 2010. So, and this is how the fields were looking like. They were completely covered with stripe rust, and these small stockholder farmers that own no more than 4 to 5 acres in average, they were having completely crop failure. So as a response of this epidemic, farmers immediately start adopting cultivar Digalo. And this cultivar has 3 main characteristics. It's a high yield crop, it's resistant to stripe rust, and also it carries SRTMP, which was a gene that is effective against TTK-SK. Because this rapid adoption of Digalo, the country was rapidly increasing its production, reaching a record high in 2012. So just in a matter of 1 year, from 11 to 2012, Ethiopia increased its wheat production in 18%. But unfortunately, this story doesn't have a happy ending. In November 2013, a severe localized stem rust epidemic started happening in the zones of Vale and Arsi. And this is one of the main wheat production regions in Ethiopia. And what was seen there, it was that Digalu was having complete failure or 100% yield loss. And this is what this picture is showing. On the right side, this is Digalu protected by fungicide having a really nice crop, when on the left side, this is unprotected Digalu having complete failure of the crop. So in response of this severe epidemic, there was an immediate and coordinated action between these three research institutions. The Ethiopian Institute of Agricultural Research in Ethiopia, the Cereal Disease Lab here in St. Paul, and the CIMMYT institution with its office in Ethiopia and also headquarters in Mexico. And these three organizations working together trying to accomplish these three steps. So at the time that the epidemic was happening, samples were collected by the pathology team in Ethiopia. The samples were shipped immediately to the Cereal Disease Lab when we performed race typing and genotyping. And finally, at the BSL-3 facility, we ran this big germplasm evaluation that include a set of Ethiopian cultivars and elite breeding lines, North American spring wheat nurseries, and the international spring and winter wheat nurseries as well, mostly aiming to identify or see the vulnerability of these materials against the race or races that were causing the epidemic. So the first thing that we observed was that the race responsible for the epidemic in Ethiopia wasn't UG99 or a related race. The race was typed as race TKTTF, and this race is phenotypically different to the UG99 in these 4 genes, and most important, the difference was in SRTMP. And as you can see, that SRTMP conferred resistance to UG99, then become completely ineffective or fully susceptible when it's challenged against race TKTTF. And this is the reason why Digalu was so susceptible in the field when the epidemic started happening. Genotypic data also confirmed that race TKTTF does not belong to the UG99 race group. So this phylogenetic analysis based on 918 SNP loss sites run at Dr. Les Seibo lab showed that all the isolates that were race typed as TKTTF, they form a clade that is completely unrelated to the races to the clade formed by the races in the UG99 group. Finally, when we, after identifying and characterizing the race, we evaluate this germplasm collection. These are the international spring wheat and winter wheat nurseries, the North American nursery, and also this group of Ethiopian elite lines. And what I want you to see here is the last column, which shows that between 20 to 40% of the lines that were resistant to TTKSK then became susceptible to race TKTTF. So this race continued narrowing the amount of germplasm that is available and effective to control these 2 relevant races. Another really good example is coming from a different part of the world. So let's move to Europe right now. So in Germany, they haven't seen stem rust during the past several decades. But all until 2013, where due to unusual high temperatures in the months of June, it resulted in a stem rust outbreak and infestations in commercial fields. And what was observed that many of the most important German cultivars were susceptible, showing susceptibility in the field, and limited resistance present in the main German cultivars. So again, we have following the same step that we did during the Ethiopian epidemic, and in collaboration with our colleagues in Germany, we followed these 3 steps. Samples were collected during the month of June in Germany. They were shipped to the Fort Detrick laboratory in Maryland. They increased the samples, and in December 1st, they shipped the samples to CDL when we conducted race typing and genotyping. And then after we identified the races, we performed germplasm evaluation, and in this case, including North American winter wheat from the International Nursery, and also today we just received 100 winter wheat and spring wheat German cultivars that will be also evaluated against the races that we identify. So the first thing we wanted to know if this unusual outbreak in Germany was due to UG99 or a related race. So did UG99 reach Europe or not? But after processing 17 stem graft samples, we identified 5 races, and none of them belong to the UG99 group. So today I'm going to be presenting 2 of the races that we think are the most relevant ones. So look what we have again. It's race TK-TTF, but in this case, the German TK-TTF is phenotypically different to the one that we observe in Ethiopia and the ones that caused the epidemic. This TK-TTF from Germany was also virulent to additional stem graft resistant genes that were not present in the differential. And in particular, it was virulent on SR7A. And SR7A is a gene that is widely present in North American spring wheat. And after screening the North American spring wheat collection, we've seen that several of these materials were virulent when we evaluate against race TK-TTF. And the second one is race TKK-TP. And this race has a unique virulence combination. It's virulent on SR24, SRTMB, and the temporally denominated 1RS amigo gene. And those 3 genes are the only source of UG99 resistance in North American winter wheat. And indeed, when we screen the North American winter wheat nursery, we find that over 50% of the cultivar that were resistant to TTKSK then become susceptible to the race TKK-TP. You can see that these races are not only relevant in the country where they're happening, they are relevant to other wheat growing regions in the world, and for example, like here in the United States. So several new races with relevant virulent combination has been identified at the CDL in the past few years. And many of these races represent a threat to wheat production worldwide. And we are not talking about only UG99 and related races. There is also other races not related to the UG99 group that can also pose a threat to wheat production. Screening national and international surplus with these virulent races provide breeders with really good information for them to define the breeding strategies for stem graft resistance in wheat. So let's move now to the second component of my research, so the identifying sources of stem graft resistance. And before, I would like to introduce you to the 2 international screening nurseries that were established in East Africa under the BGRI and DRRW project. The first one, and maybe the most well-known, is the one located in Njoro, Kenya. This is a nursery that is focused on bread wheat and uses as inoculum races in the UG99 group. The second one is located in Debre Zeit, Ethiopia, and it's a nursery that is focused on durum wheat. This nursery is located in a major durum wheat growing region in Ethiopia and uses as inoculum, in addition to UG99, a group of local isolates that are highly virulent on durum wheat. So back in 2005, when North American durums were evaluated in Njoro, it was observed that this material has a higher frequency of resistance to UG99 than North American bread wheat. And we postulate that this resistance to UG99 was due to SR13. And SR13 is one of the main components of stem graft resistance in North American durum wheat, also in CIMIT durum wheat, and it's a gene that is highly present in Ethiopian cultivars and land races. So when this resistance selection, when they're evaluated in Ethiopia, most of them became like this, completely susceptible. So we hypothesized that at Debre Zeit there might be stem graft races that possess a virulence combination that can overcome the TTKSK resistance. So when we sent our materials in 2009 and went to Ethiopia to score the nursery, we also were interested in identifying which was these races that were causing the epidemic. So we collect samples from the nursery, ship it to the CDL, and we process them here in St. Paul. So in addition to UG99, which was a race that we knew was present as inoculum, we identified these 2 relevant races, race TRTTF and JRCQC. And these 2 races, you can see, has the particularity that they have a virulent combination of SR13 and SR9A. And these 2 are the main component of stem graft resistance in North American durum wheat. In particular, JRCQC appears to be a race that is highly adapted to durum wheat. And you can see as SR13 is effective against UG99, it became susceptible when it's challenged against races TRTTF and JRCQC. So from over the 50 stem-resistant genes that has been identified and characterized, only a limited number of them came from tetraploid wheats. Most notably, SR2, which is the most widely used and best characterized adult plant-resistant gene. But the remaining ones are not highly effective anymore. So it's definitely a need to incorporate new stem-resistant genes to broaden the basis of resistance in durum wheat. So when we consider the wheat gene pool, we can see that it's composed by over 50 diploid, tetraploid, and hexaploid triticum and echelop species. And all these cultivated and wild forms can contribute with new resistant genes that can be incorporated in the modern cultivated form, either bred or durum wheat. If we consider only the tetraploid wheats or the turgidum group, we can see that there are 5 subspecies that compose them. There is only one wild form, which is wild emmer. Wild emmer is progenitor of cultivated emmer, and then cultivated emmer is the progenitor of these 5, what I call the 5 cousins. The only modern form, which is durum or pasta wheat, and these 4 ancient cultivated forms, Persian, Polish, Oriental, and polar wheat, which are still grown in some regions in the world, including Ethiopia, some countries in Middle East, and also East Europe. At the National Small Grain Collection here in the United States, there is a large number of tetraploid wheats, and that includes about 8,300 durums and 2,100 other cultivated and wild tetraploids. And our objective here was to identify and characterize new sources of resistance in this entire turgidum collection and elucidate the genetics of resistance and mapping of resistant genes. So this table shows a summary of the species and the number of entries that we characterized in the past 7 years. And you can see that our main focus is being in durum and cultivated emmer when we perform both adult stage and seedling evaluations, where for the remaining species, we focus only on seedling characterization. So overall, we are about 9,000 of the entries deposited at the germplasm bank that has been fully characterized. And this is the approach that we have been following for phenotyping the durums and cultivated emmers. So all these entries are first characterized in the field in Debre, Ethiopia. But don't think that these 7,000 entries will go all together to the Ethiopian nursery. I will be dividing this collection in groups of 1,000 and send one at a time in every growing season. Whatever lines were resistant to moderately resistant in this first field evaluation, then is evaluated 2 additional growing seasons in Ethiopia and 2 growing seasons here in St. Paul. And after 5 field evaluations, the lines that remain resistant to moderately resistant, then they are characterized at the seedling stage. Starting with these 3 relevant races, UG99, the TRTTF, which is the one with the 13 and 9E virulence combination, and race TTTTF, which is the North American race with the broadest virulence spectrum. Entries resistant to TTKSK, then they are further characterized against these 3 international and 6 North American races. So in the field, in Debre, the first 2 years, 2009 and 2010, we were using as inoculum the one that was already present at the nursery, which is a 50% of UG99 and the other 50% a bulk of local isolate. But after 2 years, we found that TTKSK wasn't highly informative for stem-bar resistant in durum wheat. So we decided that since 2011, in our portion of the nursery, we will use only the Ethiopian isolates. In St. Paul, we use all these years this bulk of 6 North American races that altogether provide us with a really good virulence spectrum. So this chart is like a summary of the result that we have seen after 5 field evaluations. And you can see that after screening 7,360 durums and emmers, see that only 5 and 10% of these entries were resistant to moderately resistant. But we can see this result from a different point of view. You can see that there are still 360 durums and 39 emmers that still prove to be highly resistant in the field and can contribute with good stem-bar resistant genes. So where are these entries coming from? So in this chart shows the origin of the resistant entries, and we can divide this group in 2 main ones. The first one are modern cultivars and breeding lines that are coming from North American breeding programs. And that includes North Dakota, mostly in the United States, CIMIT in Mexico, and Canada. And we have a second group here, which are entries coming mostly from Ethiopia and Egypt. But in this case, we are talking about land races or very old cultivars. And I'm sharing this slide with you because I've been having a very fluent conversation with the CIMIT durum breeder, and he's definitely interested in selecting some of these materials and incorporate them in his future crossing blocks to improve the genetic stem rust resistance in CIMIT materials. And also in Ethiopia, they're starting a project of exploiting local land races, not only for stem rust resistance, but also for other traits. And they are definitely decide to incorporate some of these lines into their breeding program. So after we performed the field evaluations, we did the characterization at the seedling stage of these resistance selections. And you can see that after screening 12 races at the seedling stage, we have two main groups. This one highlighted in purple, which are lines that were resistant in the field and also resistant across all the 12 races at the seedling stage. And these are definitely a group that can contribute with broad spectrum seedling or major genes resistance. And this group highlight in orange are lines that were mostly moderately resistant in the field and then susceptible across all the seedling screening. And these lines are definitely good candidates for contribute hopefully with new adult plant resistant genes. So these are like the infection type that we have seen in both durum and emmer wheat. And for all the tetraploids following the 0 to 4 scale developed by Stackman, tetraploids mostly will be ranging in the infection type 2, going from like the 2- all the way to 2+. But we have seen also entries that show a high level of resistance, and in some cases almost reaching immunity. So this table is a summary of the evaluation that we performed from the other cultivated and wild tetraploids. And you can see that in this case, after screening 10 races at the seedling stage, again, it's only about 5% of them that were resistant across all the races. So wild emmer, and not surprisingly, is the subspecies that showed to have the highest level of diversity for stem rust resistance, and they are definitely the best candidate to incorporate stem rust resistant gene that has not been used in agriculture. Also, polar wheat shows a high frequency of stem rust resistance, and it's another cultivated form that can contribute with good stem rust resistant genes. So after we complete the phenotyping, then we move to the second part, which is the study of the genetics of resistance and mapping. And for that, you can see that I perform about 50 biparental crosses, mostly focused on durum wheat, but also include the other tetraploid species. I advanced to the F2 and F3 generations, and both generations were phenotyped at the seedling stage against UG99 and the TRTTF race. And we are following the bulk segregant analysis to map these stem rust resistant genes using the 90K SNP platform that is running Fargo at Xiaoman's Xiao lab. So as a brief summary, in the genetics of resistance, we found that resistance to UG99 in tetraploids is mostly conferred by one gene, and in some cases, two genes. And we have seen both dominance and recessive inheritance. Very interestingly, that in many of these durum parents, when we evaluate against race TRTTF, we were able to identify additional stem rust resistant genes. So in other words, some of these durum lines that have only one resistant gene effective against TTKSK then prove to have 2 or up to 3 genes that were resistant against race TRTTF. And these genes can contribute to broaden the basis of stem rust resistance, not only in durum, but also in bread wheat. A really good example of how this resistant selection has been utilized is this collaboration project that we have with Stephen Yu in North Dakota. And what Stephen has done was he selected 46 of the resistant emmers that we characterized at the field and seedling stage, and he developed synthetic wheat by crossing the emmers with resistant and susceptible Aeschyloptausia, which is the donor of the D genome of wheat. So this synthetic definitely can provide with a great source of unexplored genetic variability for wheat improvement. In particular, this cross that combines the resistance that is coming from the tetraploid with the resistance that is coming from the diploid species. So in addition to the tetraploids, we have also been exploring the tertiary symphyl of wheat for resistance to stem rust. And you can see in this table, we characterize 9 diploid and tetraploid aeshilop species. And we perform an evaluation for about 8 races, but today I'm presenting a summary to the 3 relevant ones. You can see here that for the aeshilop, the frequencies of resistance are much higher than the one that we've seen in the tetraploid wheat. So here reaching almost 40% of the entries. And as this picture are showing, many of these entries showing a very high level of resistance, in some cases almost reaching immunity. So there is a lot of potential here also to get new stem rust resistant genes. Same as with the tetraploids, with the aeshilop we have developed several mapping populations, and with the objective of first understanding the genetics of stem rust resistance, then mapping of resistant genes, now focusing on diploid aeshilops using the genotyping by sequencing approach. And this is an effort that Erena Adae is leading at Matthew Rau's lab. And then also to generate genome-wide genetic linkage map that will allow to exploit these wild relatives, not only for stem rust resistance, but also for other traits that can be contributed from these wild forms. So finally, I would like to spend a couple slides of the education component that result from this international research collaboration that I have been involved in the past few years. And the first one, as Matthew said in the introduction, is happening here in St. Paul. So back in 2010, I was offered to develop and implement a training program on cereal rust diseases. And this program is under the aegis of the Stagman-Burroughs Center of Sustainable Plant Health. It's a 6-week summer training program that is focused on cereal rust prevention and control, and is targeted mostly to international scientists. And several faculties, postdoc and students that are present here right now have been contributing with the time to train these international scientists. So this program has been training pathologists and with scientists from Ethiopia, Pakistan, Nepal, Bhutan, Georgia, and Kyrgyzstan. And one trainee from Pakistan is sitting here in this seminar today. And in addition to what I would like to mention, that in addition to the education opportunity that this training was providing to these international scientists, the possibility of having them visiting campus has strengthened also the research relationships that benefit the department, the cereal disease lab, and also the research institution where these trainees were coming from. And also in Ethiopia, in particular the past 3 years, I have been invited as a resource person for participating in hands-on training to scientists from different national research centers. And this was the last one that happened last October, and you can see that Ruth Dilmaki was also present there, covering Septoria and Fusarium head blight diseases as well. And also I've been heavily involved in assisting in scientific capacity building, including provision of laboratory equipment, mostly at the AMBO Plant Protection Center. So I was working with the pathology team in AMBO to establish a laboratory that is able to conduct race typing and surplus evaluation. So in summary, wheat stem rust, it's a re-emerging disease of wheat, and it's again posing a threat to wheat production worldwide. Fortunately, international research collaboration is happening, and it has been essential for making substantial progress in prevention and control of stem rust. Detection of new virulence in the pathogen and monitoring spread, and finding new and diverse stem rust resistant genes has been our contribution from St. Paul to this big international effort. So I would like to close saying that all the results and the research I present result from a big team collaboration happening here in St. Paul, and also with a big group of international collaboration that make this research be successful and being able to provide with really good research results. So with that, thank you very much, and I'm open to all the questions that you have.